Alright, welcome to your video of the remainder of Lecture 7. For our last clicker question, we were looking at this gene that has 3 different alleles for putting antigens on red blood cells, and I asked you which of these 3 alleles, which I've described here, are the codominant ones, and the correct answer was I with the big A and I with the big B. So the reasoning for that, or the logic you could have used to figure out the answer, number one is that I did give the capital I to both of these alleles, so that kind of gives you a hint that they may both be dominant, therefore codominant. I also used the superscript A and B, so this notation usually we use when we can't tell which one is dominant, for say incomplete dominance or in this case codominance. So the symbols could give you a clue. But also because it kind of makes sense, right? It makes sense that if you have one allele that makes one type of protein and another allele that makes another type of protein on the red blood cell surfaces, that if you have a heterozygote with both of these alleles, you'll end up getting both of these types of protein. So in terms of the antigens that are found on the red blood cell surface, it makes sense that a heterozygote will show both phenotypes fully and therefore be codominant. So codominance means that when you have both alleles, both of their phenotypes are fully expressed at the same time. So why is all this kind of very important and interesting to you? Well, a bunch of reasons. One is that the genotype that you have determines the antigens that you have on your red blood cells, as we have already discussed, but why it matters to you is that that also determines your blood type. So if you have type A blood, what type of genotype do you think you could possibly have? All right, and also which antigens would you produce? Okay, so that's a question to kind of help you think about blood types and the genotypes that would produce those blood types. All right, so pause the video now and decide on your answer, and then come back and then pause it when you've committed to a decision. Okay, welcome back. So the correct answer is B, right? So to have type A blood, it means that all you have on your red blood cells is the type A antigen, but there's 2 different genotypes that will give you that type of blood. One is if you have 2 copies of the A allele, or if you have 1 copy of the A allele and 1 recessive small I allele as well, the one for type O blood, because that doesn't produce any antigens at all, doesn't affect the fact that you're making only type A antigens. Okay, so remember that the small I allele is small I because it's recessive. It's fully recessive to both the A and B alleles, and it makes no antigens at all. Okay, so if you're big A and small I, or if you're, you'll still only make antigen A, and therefore have type A blood. All right. So second reason why this is important and interesting is because your immune system actually can recognize what type of antigens are normal for your body, what type of antigens should be there on your blood. And anything that shouldn't be there will be attacked by your antibodies. All right. So for example, if you have type A blood, right, what types of blood do you think you can accept for blood transfusion? So if you were injured, you lost a bunch of blood and they have to give you someone else's blood, which types of bloods can you accept into your body? All right, so pause the video and choose your answer. Okay, welcome back. So if you chose E, then you are correct, right? If you normally have type A blood and your body knows that, oh, seeing this type of antigen A on my red blood cells is totally normal, then you can accept other type A blood because it looks exactly the same as yours. Or you can also accept type O blood because there's no antigens there at all. As far as your immune system is concerned, this is invisible. There's nothing new that doesn't belong, and therefore you can accept type O blood as well. So notice that, however, that you cannot accept type B blood or type AB blood, because your body's going to see this spiky B antigen and say, what the heck is that? That does not belong in our body. That must be some sort of virus, bacteria. Kill, 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 right? So that'd be very bad. You'll be attacking the new blood that you need, and your body would suffer a lot from that. Okay, so you can't take any blood that has type B antigens on it, on its red blood cells. Okay, so that's kind of why people, the blood bank is always asking for type O blood people, because they are universal donors. Their blood doesn't have any antigens at all. They can go to anyone, right? So they're universal donors. So with no antigens, nobody's immune system is going to attack this type of blood, and therefore it's the perfect blood for everybody, right? But it also is why type AB blood people are universal acceptors. Like, to someone with AB blood, right, any type of antigen is normal, right? So type B blood comes in and says, oh, okay, that looks kind of normal. Type A blood comes in, oh, that looks normal as well. Type O blood, oh, I don't see it at all. So these are universal acceptors. They can accept blood from anyone, all right? So both types of antigens are normal for them, so they can accept any type of blood. So it's kind of opposite. Being a good donor, type O blood, makes you a very picky recipient though, right? Because your blood normally has no antigens at all. You can't accept blood from anybody else except for type O person, because to you, any sort of antigen is wrong. It doesn't belong, right? Whereas a type AB blood person, they are very good acceptors, but they make very poor donors because the only other people that can take AB blood without a bad reaction is other type AB people. Okay, so pretty interesting stuff. Third reason why this is interesting is because this is another one of those kind of easy biological parent type tests. Also another risky one for us to talk about in class in case you're discovering something new about your family. So apply this logic to your family at your own risk. And do remember that other mutations and genes can be involved and that your parents do love you. Maybe the blood tests were wrong. Okay, anyways. So thinking about the possible genotypes of you and your parents, right, or basically of any father and mother combination producing a child of a particular blood type, right? Which of the following combinations are, let's just say, most biologically probable? Okay, so pause the video, take a look at each of these examples, and pick the one that is most likely to happen, is the most normal. Okay, pause it now. Okay, so welcome back. So choice C is the one that's most normal. Okay, because it's possible that this person with type B blood is actually carrying a recessive kind of O allele as well, right? The person with AB blood has to have both, has to have a big A and a big B in order to have type AB blood. But with this parentage, it's possible to get the big A from this parent and the small I from this parent and have only type A antigens. Okay, whereas all these other ones are not possible. For example, this one, there's no B alleles present at all, and you couldn't have type B blood. Okay, so you can go through each of these examples to see which ones, why they don't make sense. Okay, moving on. So far, we've been looking at just single genes at a time with with that last example, 3 different alleles. And we looked at examples of incomplete dominance and co-dominance. Next, we're going to look at genes that have way more than just 2 or 3 alleles. Okay, so more multiple allelism. So the fourth example is going to be looking at an allelic series of 4 fly eye color. So we looked at Drosophila fly eye color before. For example, we looked at one that we only looked at 2 alleles before for an eye color gene. For example, we looked at one where the wild type allele produces kind of the dominant, really dark purplish red kind of wild type eyes, whereas the recessive allele is a mutant where you don't make the pigment at all and therefore have white eyes. Okay, so 2 different alleles that we looked at before. But in reality, this gene actually has a whole series of different mutant alleles. So it's still one gene, but there's a whole bunch of different versions that exist. For example, there's one called, for the white gene, that we call the cherry allele because it gives you this more reddish cherry color. And we call that a weak mutant because it doesn't really affect the color that much. It just makes it a little bit lighter, a little bit redder. Okay, so we call it a weak mutant because the mutation doesn't really damage things or change things too much. There's one called apricot, which is quite a bit lighter, more kind of pale yellowish orange, and that we would call a stronger mutant because this is more different from the wild type, from what's normal. Okay, so this is a more severe effect on the phenotype when you have on this mutation. Okay. So when we say strong mutant, we mean it looks less like the wild type, less like the white wild type allele. Okay. White 1, the one we looked at before already, we could call it the strongest mutant because it's so strong that we have no color at all. It's completely kind of defective. It's so severe that you're unable to make any pigment at all. Okay, so kind of wild type, and then a weak mutant is a little bit different, stronger, even more different, strongest one, totally different. Okay. So we call this white allele the null mutant because it's the one that is completely non-functioning. Okay, so remember that terminology. We may mention it later. Okay, so let's talk about, so we have a gene that has many, many different alleles. So what happens when you have 2 different ones together? So when you have 2 different alleles, when you're heterozygous, right? For this allelic series anyways, one allele is always completely dominant over the other, all right? So if that's the case, right, and knowing what you know about these alleles and how they might affect the genes, right, how might you put these alleles in order from the most dominant to the most recessive. I guess technically that should say alleles, not genes. Okay, so pause your video and choose your answer. Okay, so hopefully you thought about it and chose A. Alright, so this is what we call an allelic series. An allelic series will list the symbols for all the alleles that we're looking at, but it'll also tell us which one's the most dominant, and the next most dominant, and the next most dominant, and the least dominant, or the most recessive one. Alright, so why does that make sense? Well, it makes sense because the data basically proves that, and we'll talk about why the data looks the way it does afterwards, right? So if we're looking at an individual that has one copy of the apricot allele and one copy of the null allele, right, we would expect the offspring to look like this, right? Because the null allele makes no pigment at all, the apricot one makes this kind of weak, kind of yellowish kind of color, right? So whichever one is the most functioning creates the color that we see, right? If we have an individual that has one copy of the cherry allele and one copy of the apricot, right, we would expect the offspring to look like this, because this one's quite defective, but this one's able to make some pretty good pigment that's almost the wild type color, so we would see this stronger color instead, this deeper color, the more wild type color. Alright, so as you can see with this allelic series, whichever one's further to the left is the more dominant one, whichever one's further to the right is the more recessive one. So cherry is dominant over apricot, which would be recessive to it. Okay, so reasoning for that we kind of just mentioned, right? The stronger the mutant allele, so as we go from left to right and go to a stronger and stronger and more severe mutation, basically the more messed up it is, the crappier it is, the less it works, right? So these weaker, less messed up alleles, right, are ones that end up performing a better job of the normal function, and therefore it gives us the phenotype we see. Okay, so the stronger the mutant, the weaker it is in terms of dominance and recessiveness. Okay, so stronger the mutation, more recessive it is. So the strongest mutant of all, the null one, is the most recessive. It's recessive to all these other alleles, because this makes nothing, this stuff at least makes something. Okay, so moving on. So in reality, we looked at kind of, how many? 4 different alleles. In reality, there's actually 12 different alleles that we've identified for just this one gene, all right? So here they are listed from the strongest mutation, the null one, the white one, all the way to the weakest, which is actually wild type, the normal one, okay? And you can list them based on the amount of pigment that gets produced, right? And it also happens to be listed from the most recessive, right? The strongest mutation is the most recessive to the most dominant, which is the wild type. Okay, so this is kind of another example of an allelic series listed in order from kind of dominant to recessive, or strongest mutant to weakest mutant. Okay, so next example. So let's take a look at another allelic series, in this case for rabbit fur color. So the rabbit fur color gene also has multiple alleles, and the allelic series looks like this, where the wild type kind of most dominant allele produces kind of the full dark color for the rabbit. The next, the weakest mutation is chinchilla, which just makes it a little lighter colored. Himalayan has a particular characteristic pattern, but a lot of it is white, and then albino is completely white. All right. So this is our allelic series with the most dominant alleles here, the most recessive ones here, the weakest mutants here, the strongest mutants, or the null mutant over here. So here's a question for you guys. So let's say that you have a rabbit named Mr. Bun, which has chinchilla fur coloration, so it looks like this. Okay, how many different genotypes can produce this coloration? Okay, so if you list all the possible genotypes that Mr. Bun can have, how many different genotypes could you have? All right, so pause the video and think about it. Okay, so hopefully the answer that you committed to is D, 3. Right, so let's list them out. So one possibility is just being homozygous for the chinchilla allele, so that would definitely produce the chinchilla phenotype. You could also have chinchilla and Himalayan, because remember, everything to the right of this is more recessive, right? So you'll still see the more dominant chinchilla coloration. Same with chinchilla and albino. The no mutant doesn't really do anything at all, right? So every one of these mutant alleles that is stronger in terms of how messed up it is actually more recessive to this chinchilla allele. Okay, so that's what the allelic series is telling us. So get used to that language in terms of strong mutation means more messed up, and therefore means more recessive as well. Okay, so the mice have a dominant disorder, so that if it has a big T allele, it will have these short, stubby tails. So this tail isn't curled around the body, it actually just stops right there. That's how short the tail is. Whereas if it has two of the recessive alleles, it would have a long, normal tail. Right? So it's a dominant disorder. The disorder is caused by the dominant allele. So the researchers wanted to study this gene, and so they performed a bunch of monohybrid crosses. So they crossed big T small t with big T small t. And in their results, what they observed was out of 3,000 offspring, 2,000 of them had stubby tails like this, 1,000 of them had normal long tails like this. But that's kind of weird, because remember from our monohybrid crosses with complete dominance, we expect to see a 3-to-1 ratio of the dominant phenotype to the recessive phenotype. So, and we have enough results that this is probably not random chance, and this is not just due to some weird luck, right? So how might you explain this unusual result? So pause the video and make a decision. Okay, welcome back. So the explanation that makes the most sense is B, right? If the big T allele is lethal, meaning that it makes the mice not able to survive whenever it's homozygous, that would explain the results, right? That would mean that all of the big T individuals, or if for some reason they only have one copy and they're hemizygous for big T, then they would give you a 2-to-1 ratio. If you look at the Punnett square here, right, normally we would expect a 3-to-1 ratio because all of these have a big T, they would all have stubby tails, 3 of them versus just one homozygous recessive normal. But if big T is lethal, if all of these individuals die before they're even born, all we'll ever see in those monohybrid cross offspring would be 2 stubby tails to every 1 normal tail. All right? So that's why we call this a lethal mutation. It's a mutation that when you have 2 of them, or at least it's recessive for lethality, when you have 2 of them, then you don't survive. Okay. So why is it that this allele, which causes short tails, when you have 2 of them, why do they die? Well, it basically just tells us that this wild-type allele, besides kind of determining tail length, also does other important things that are needed for the mouse to survive. So if you have 2 messed up copies and you don't have a normal small t at all, then you're not able to survive. Okay, so this one's too messed up, and it's too important of a gene to be lacking one, at least to not even have one good copy of the allele. So these are lethal mutations. We'll talk more about these later on when we're talking about 2 genes. Okay, but here's another example. So achondroplasia, so we're still looking at lethal mutations, but a different example of it. So achondroplasia is a type of dwarfism, and it's caused, again, by a dominant allele, so big D, right? So big D individuals don't survive, though. It's also a lethal mutation. So it's dominant for causing dwarfism. It's recessive for lethality. Okay, this kind of makes sense. Now, this one actually is a little bit easier to understand than the mouse tail one, because it makes sense that if just having one bad copy of this D gene causes such dramatic effects on the growth and development of the body and on the morphology and the health of the individual, then having 2 copies of it would cause way too many health issues for the offspring to survive the pregnancy event. Okay, so this is an example of pleiotropy. So is the mouse example. Pleiotropy is when one gene ends up affecting 2 or more traits that are quite different, 2 more phenotypic traits. So this gene for the dwarfism phenotypic trait, it's dominant for dwarfism and it's recessive for normal. Okay, for normal height. So small d gives you normal height. Big D small d makes you a little person. But if you're looking at the other lethality side of things, you could call it the death trait, right? The big D allele is actually recessive, right? Because when you have 2 of them, you wouldn't be alive, all right? Let's look at another example of pleiotropy. So sickle cell, right? So when it comes to having anemia, right, lacking the ability to carry oxygen very well in your blood and therefore being quite weak and lacking oxygen all the time, out of breath, you could argue that the gene, the mutant allele is recessive when it comes to the symptoms, right? In that you have to have 2 bad copies in order to show the full symptoms of sickle cell anemia, all right? But you could also argue that for sickle cell gene, it is codominant in that when you look at the blood, a heterozygous individual has both normal and sickle-shaped blood. And so you can say that both the normal and the sickle mutant alleles are expressed in their phenotype fully, right? So sometimes whether it's co-dominant or whether it is recessive depends on what you're looking at, what phenotype you're focusing on. The whole body's reaction to its genes, or in a finer scale, what types of blood cells you see, okay? But it's pleiotropy because besides affecting blood cells and therefore, the functioning of the body in terms of oxygen carrying, it also affects how you react to malaria, right? So your malaria symptoms are affected in a rather strange way, in that if you're heterozygous for the sickle cell gene and you have kind of both types of cells in your body, you actually suffer the least from malaria, right? So you actually handle it even better than someone who is wild type and has all normal red blood cells. Maybe just because these sickle cells or the proteins themselves make it more difficult for malaria to take hold and to damage your body, right? But if you're homozygous for the sickle cell allele, and maybe because you're already suffering from this severe anemia, malaria affects you even more strongly. All right. So the other side, the pleiotropic side of the sickle cell gene, where it's affecting your malaria susceptibility, is interesting in that heterozygotes do great, homozygotes do bad. Homozygous for anemia does bad. Homozygous for normal does kind of in between. Right? So it's a little bit more complicated, but the key point from this is that we see two different traits. So susceptibility to malaria and your red blood cell type and amount of anemia are affected by one gene. Okay, that's what we mean by pleiotropy. Now, another thing about these lethal mutations is that you can't make a pure breeding line. So as much as I said before that you want to start with pure breeding parents when you're setting up crosses just so you know exactly what you're making, it's impossible to do that for something that has recessive lethality, right? Because you can mate two little people together, right? And as a result, you'll get two little people for every one normal height person. But you never get a big D because these individuals don't survive. So if you can't make big D, you can't make a pure breeding chondroplasic population, right? So next thing I want you to think about is, to pause the video and think about is, what other phenotypes can we not make a pure breeding population for? Okay, so pause and think about that for a sec. Okay, welcome back. So any phenotype that appears only in heterozygotes is another type that you can't make a pure breeding population for. All right. So for example, for incomplete dominance, right, you can't make a pure breeding pink flowered carnation line, and you can't make kind of these a pure breeding line that is always tortoiseshell or kind of red and white, because these are all heterozygotes, right? So even when there's no lethal alleles involved, there are still phenotypes such as these that you cannot make a pure breeding line for because they are not pure. They're not made from homozygous genotypes. Okay. All right. So one last kind of example of lethality and pleiotropy: mouse coat color. Okay. For mouse coat color, big A gives you the wild type agouti color, which is the darker ones you see here, and the big A with the Y mutation gives you the mutant yellow color, which is the lighter ones you see here. Okay. So the yellow, the AY mutation is kind of dominant for coat color. All right, so when you have AY with A, you end up seeing this lighter coat color, but it is recessive for lethality, meaning if you have two copies of it, then you wouldn't survive. Okay, so once again, if you're heterozygous, AY is dominant in terms of fur color, but if you are homozygous for this dominant allele, you would get pretty much a dead embryo. Okay, I know this is a lot of examples, but I bring this up because your mastering genetics ends up asking you questions about this specific example. Okay, so you should know it for that reason. All right, example 7. So environmental effects. We've been talking about genes as if that's all that matters, that the genes you have determine exactly who you are, but that's not the case. Environment matters as well. So one example of that is how temperature affects your phenotype. So when you look at these animals that have what we call Himalayan coloration, right, where there's these kind of dark parts and lighter parts, right, would you think that the darker areas are hotter or cooler than the light parts? So pause the video and think about that for a sec. Okay, we'll come back. So hopefully you figured out that the darker areas are actually the cooler parts of the body parts that are more on the extremities and therefore can kind of lose heat more and be cooler. Alright? So considering that the darker areas are cooler, the areas with more pigment are cooler, right? What does this tell us about this mutant allele that gives us Himalayan coloration, right? So how does this mutant allele give us this temperature-dependent pattern? All right, so pause the video and think about that for a second, see if you can come up with an idea. All right, welcome back. So the way these alleles may work is that, or the way they do work, is that when the temperature is cool enough, right, what we call a permissive temperature, this mutant protein is still able to fold properly and therefore make the darker pigment. Okay, so in these cool areas, the protein can fold properly, can function normally. But at temperatures that are higher and are therefore too hot for this mutant protein, what we call restrictive temperatures, then these proteins, which are mutated, they're slightly unstable and their temperature unstable, they don't fold properly, they can't do the jobs of making pigment, and therefore you see less or no pigment being made. Alright, so basically this mutation basically damaged the allele in such a way, that the protein it makes is not temperature stable. When it gets too warm, it doesn't work. When it's warm enough, and when it's cool enough, it does work. Okay, so that shows you kind of the mechanism behind how the environment affects the phenotype. Okay, here's another example of temperature affecting phenotype. So many plant phenotypes are very temperature dependent as well. As a very clear and interesting example, there's a plant where having two big R alleles gives you red flowers, having two small R alleles gives you ivory flowers, but if you're heterozygous, something kind of interesting happens. When it's cool and bright and you're heterozygous, you end up making red flowers. When it is warm and shady, you end up making ivory flowers. So this kind of complicates your genetic analysis. It's a good thing that Mendel didn't choose to use this plant, because how you interpret the results depends on the growing conditions. What you see in this next generation depends on how cool and bright or how warm and shady it is, right? All right, so examples 8 and 9, we're going to talk about expressivity and penetrance. Okay, so two more different kind of twists to Mendel's genes. So genes are not always expressed as kind of 100% completely and consistently as you might expect. For example, all of these individuals, you could say, have the same genotype when it comes to polydactyly. Okay, so they both have one normal and one mutated allele, but because this is a dominant disorder, they will express polydactyly. But notice that this degree of polydactyly varies a lot, right? Polydactyly meaning you have extra digits, right? So this one here, for example, just has a little bit of an extra thumb. This person has an extra thumb but a small one. This person has one extra finger, two extra fingers, right? Half of an extra toe, one extra toe, more than one extra toe, right? So same genotype, but very different expression, different amount of kind of this phenotype being seen. So this is what we call kind of differences in expressivity. So the degree to which this mutant character is expressed, the amount of extra digits you see, they do vary between individuals. All right. So even though they have the exact same two alleles, the varying expressivity tells us that you can express that gene to different extents. Okay. Also, sometimes a person can have a dominant mutant allele, such as the one for polydactyly, and don't show the mutant phenotype at all. They can have kind of normal hands, normal feet, same number of 10 fingers, 10 toes, right? So they look totally wild type. So this is what we call incomplete penetrance. So it's almost like you have the gene, but the gene's phenotype didn't penetrate to be shown in the surface, kind of thing. So the way we can kind of help you understand the numbers behind this is, if you have complete or 100% penetrance, that would mean that everybody who has this dominant allele will show at least one extra finger or toe, right? You'll see something, right? But incomplete penetrance, so let's say you have 95% penetrance, that would mean that 5% of the people will have the mutant dominant allele, but totally appear wild type. Okay, so 95% show it, 5% don't. Okay, that's what we mean by penetrance. All right, so to help this terminology sink in, I've made kind of 3 populations here, all of which should have the dominant mutant allele for purple color. But I want you to kind of look at each population, considering they all have the same genotype, and tell me whether expressivity and or penetrance is varied. okay, is variable. So in each group, is expressivity varied, is penetrance varied, or is both varied? Okay, so pause the video and decide which terms apply to each of these groups that have the same kind of dominant phenotype. Okay, pause now. Okay, welcome back. So in this first group, I would say that only penetrance is varied because not everybody expresses a mutation. So these people express the purple mutation, these other ones don't at all. But the ones that do express the mutation show the exact full phenotype, so there's no differences in expressivity. All right. Whereas this next group, I'd say there's variable expressivity, but not variable penetrance, because everybody's showing some amount of purpleness, so shows some amount of the mutant phenotype, but some to a lesser degree than others. Okay. Whereas this last one, we have variable penetrance and expressivity, because not everyone shows the trait. Some of them are not purple at all, and the ones that do show purple show a variability in how purple they are. Okay, so hopefully that clears things up. Okay. The thing about incomplete penetrance especially is that it can make genetic analysis difficult because you don't see a consistent pattern, because where you would expect to see a mutant phenotype, it's invisible. It looks wild type instead. Okay, variable expressivity not as big of a problem because, say for polydactyly, as long as there's one extra digit or more than that, you can still know that there's a mutant allele there. Okay, so just to show you an example, when you're working on a family pedigree, if there's incomplete penetrance, you can get confusing or misleading results. For example, here's a tree for polydactyly, where you would expect this individual to be heterozygous, right? In order to pass on a dominant disordered allele to these two offspring, one of these people have to have the dominant allele. And it looks as though it should be this one because they have a family history of it, but because it's not expressing the trait because of incomplete penetrance, analyzing this tree would make things very confusing. Okay, now when this is the case, I'll tell you, so don't overuse this in your pedigree analyses. I will tell you if incomplete penetrance is an issue for a tree. Okay, so let's look at one more example of allele that shows both variable expressivity and incomplete penetrance. Okay, so the S gene, when you're looking at the P allele, right, is one that codes for having white patches of fur in dogs. Okay, so if you have this allele, you would have white patches for beagles. But if you look over here, you can see that the amount of white patches varies a lot, and in some individuals there'd be no white patches at all. So this shows variable expressivity in terms of how much the whiteness is there, from a little bit just at the feet to most of the dog being white, but also incomplete penetrance, and that some of these individuals would have no white patches at all. Okay. All right, examples 10 and 11, we're going to look at sex-influenced and sex-limited traits, and I think this is our last set of examples. Okay, male pattern baldness. Okay, if you look at a family pedigree for male pattern baldness, you see that it's a lot more common in males. Why is that? Okay, so pause the video and think about that for a sec. Okay, welcome back. So if you look closely at this pedigree, you'll see that it's not Y-linked, right? So that's not the explanation for why male pattern baldness is more common in males. We know it's not Y-linked because we see some females do have it. Okay, it's also not X-linked though, because notice that it can be passed from fathers to sons, right? So that's not something that you should be seeing so commonly for an X-linked gene, because normally the father would only pass the Y chromosome to his sons, and therefore what the dad has in terms of X-linked genes would never be passed on to the sons, be passed on to the daughters instead. Okay, so it doesn't seem to be X-linked either. So if it can't be Y-linked and it can't be X-linked, then it must be autosomal. But somehow it's more common in males. So the reason for that is that this gene behaves in such a way that when you have the male pattern baldness allele, just one copy of it, it's dominant if you're a male, right? So whether you have one bad copy, right, plus is wild type, so this is no baldness at all, this is the baldness allele. If you have one bad copy, you'll be bald. If you have two bad copies, you'll be bald as well. Okay, but in females, it's recessive, right? Only if you have two bad copies of this mutant allele would you have male pattern baldness as a female, which is why it's so uncommon here. So the only way in which this female has male pattern baldness is if she got one copy from dad and one copy from mom, right? But it was recessive in this mom because it's recessive for females, whereas for males, you just get one bad copy and you have male pattern baldness. Okay, so these are what we call sex-influenced genes. Okay, they're not sex-linked. They're not found only on one sex chromosome versus another, but they are affected by sex. Okay, so the reason why male pattern baldness happens to be affected by sex is because it's related to a sensitivity to the presence of dihydrotestosterone, and dihydrotestosterone is just present in a higher concentration in males. Okay, therefore we're kind of just more sensitive to this allele. Now there are other genes that are completely sex limited, so that's something different. It's not just influenced by sex, it's kind of only one sex can have it. Okay, only one sex can show the mutant phenotype, even though it's an autosomal gene. Okay, so once again, this is not only one sex showing it because it's Y-linked or something like that. It's because it's an autosomal gene, but only one sex can show it. So can you think of any examples of genes that only one sex can show and not the other, even though it's an autosomal gene? Okay, so pause the video and think about that. Okay, welcome back. So here's some examples. So beard growth patterns, right? Generally speaking, only males can grow beards, and so if you're a female and you have particular alleles for, having a full beard versus patchy beard, you're not going to be able to see those phenotypes. Same for impalas, the horn shape, right? Both the males and females probably carry different alleles for the shape of the horns, but since the females don't grow horns, you won't see those traits either. Okay, so this is a sex-limited trait, limited to males only because only males grow horns. Same goes for chicken comb shapes, because males have more kind of complex comb shapes. So these are all different traits that are sex-limited to males because females don't grow these particular structures. Okay. There's also other genes that are kind of sex-limited to females. For example, genes that affect lactation, menstrual cycles, probability of getting breast cancer and such. These are generally only found in the female sex. Now males do have these genes too. These are autosomal genes. Males have as many copies of these genes as females do, but they usually aren't expressed because males generally don't have breasts. Not always the case. One of my friends genetically does have breast tissue even though he's male. There's another interesting exception. So besides my friend who has a mutation that causes him to have breast tissue, strangely enough, that friend is very curiously interested in breastfeeding his own children, and he has a kid now, so I'm wondering what's going on there. But anyways, another interesting example is this. So Laura Ray, who's a really great comedian and the artistic director for the CBC, she has for the last few years been openly sharing her experiences of transitioning from male to female. So she's a transgendered female. So she talked about undergoing hormone treatments and how it's changing her body and her moods and her emotions. And when they're talking about breast size, the doctors told her to expect that her natural breast size that she'll develop will be about one size smaller than for the woman in her family. So there is kind of genetic basis for how big she can expect her breasts to grow. So in her case, right, even though she was born male, her sex-limited traits, since she is transitioning to female with the female hormones she's taking, she's now expressing some of the female sex-limited genes, right? So her genes for breast size, which she got from her family, from her parents, are now being expressed for the first time, and this is happening at age 53. So she's making this transition at a very late stage in life. All right, so that's pretty much it for lecture 7. So do go back and review this lecture with the slides or with this whole video, just to make sure you understand the patterns and the interactions, and not just kind of memorize the patterns, but know the genetic basis for it, and kind of understand all these different allele combinations and interactions you can see with just a single gene. Make sure you understand it well, because starting next week, we're going to be studying interactions between 2 or more genes, and that's going to make things a bit more complicated. All right, so thanks very much for listening. I will see you all next week.